G'day guys, how you going? So what I'm going to try and do in this RPA tutorial, sort of exam question sessions, is we're going to try and take you through a few of the questions related to the RPAs and how we'd run through them. And I'll run you through my thought process. This will take a bit longer, but I want to sort of run you through how I approach these sorts of questions so that you can take some of those skills into your own exams. I'm going to start with a key question. The key question is like what you have to be able to do, and then it'll blow out and it'll be a bit longer, and I'll do an extra question for those of you that are interested. But if all you're looking for is just the core for a 4, listen for the key question and then you could stop in the second part if you want. All right, so let's look at this in terms of our Enzymes RPA. So the first question here says, enzymes are used in biological detergents. Name the type of enzyme that digests the stains found in fats. Now we know that the fancy name for fats is lipids, okay? And we know, if you think of all the enzymes you know, amylase, protease, carbohydrates, all of these end in an ASE. And the secret is to basically have the ASE and write the first letters of whatever it is that you've got in front of it, and then carbo-high, and then you could just literally add the A's on the end, right? So if you've got a protein, it's protease. If you've got a lipid, it's lipase. And if you've got a carbohydrate, it's carbohydrase. So in this case, we've got a lipid. So the enzyme is going to be lipase, right? Done. Bob's your uncle. 1 out of 1. Killing it. Okay, so now this is probably the meat of the question, and this is kind of linking straight in to the RPA that we've done. So a new detergent is marketed as being environmentally friendly. Scientists compared the performance of this new detergent with an existing one. They measured the time taken by the two detergents to remove the stain at different temperatures. So the time taken here is on the y-axis and the temperature is on the x-axis. We've got two detergents, the existing one or the new detergent. All right, so the existing one's the checkered line, the new detergent's the solid line. All right, so describe the effect of increasing the temperature on the time taken by the existing detergent to remove the stain. All right, so the first thing I've observed here is to describe question. And a describe command word just means you need to basically see that shape in the graph. You need to take this shape and explain it in words, describe it in words. That's all you need to do. But if you're a describe, you want to make sure you're using units from the graph as well. So put some temperatures, put some times in it, things like that, okay? All right, so here we go. Now, before the effect of increasing the temperature on the time taken by the existing detergent. Okay, so we are only interested in the checkered line. We're not doing anything with the solid line at this point. Again, another skill that I use a lot of. I will underline the things that I want to make sure it draws my attention to what the question is asking. And lucky last, before I go into anything with a describe question, I cast my eyes down here and I look at this number. So this is the describe question worth 2 marks, which means they want 2 points out of you. That's what they want, okay? All right, so how can we break this line into 2 things? Well, different ways. I'm going to let's start with this point right here because it's probably one of the quite obvious points. You can see that the graph kind of comes down. Here we go. And then it goes back up after that. And there's this point which is like the top of the mountain, even if it's upside down, right? And this is 40 degrees Celsius. So it gives us a good chance to use those numbers. And this is the time taken to remove the stain. So at 40 degrees, it took the least time to remove the stain, which means the enzyme was working the best. And if the enzyme's working the best, that's what we call optimum, right? So 40 degrees Celsius was the optimum or best temperature. And I'm going to write shorthand here just to save a bit of time, guys, for the enzyme, right? There we go, 1 mark, okay? We have basically seen 1 point and observed it. Tick, great. So let's look for that second point. This second point is going to be a little bit two-sided because we've got this pattern here where as temperature increases initially, the time taken to remove the stain decreases. But after 40 degrees, as temperature increases, the time taken to remove the stain increases, right? So we've sort of got to get both of those observations in this second point. So initially, as temperature initially, excuse all my really poor spelling, initially as temperature increases, as temperature increases. There's a little arrow there saying increases. What's going to happen is the time taken to remove stain decreases, right? Stain goes down or decreases. Again, I'm just drawing arrows just to make it a little bit quicker. After, and you'll notice that I'm using these numbers, right? After 40 degrees Celsius, however, as temperature increases, time taken, and you repeat it out again, time taken to remove stain, now it increases because it's going back up, isn't it? Increases. All right. Boom. Done. We've got 2 points, tick, tick, for that question done. And break it down that way. Highlight, underline, do whatever you want to this paper. Scribble on it. Look for these things like the command words and the marks for the question. Really important things. Okay, so let's have a look at this next one. So the new detergent works at a lower temperature. Is the new detergent likely to be more environmentally friendly than the existing detergent? Draw a ring around your answer. Well, it is, okay? Explain the reason for your answer. So yes, it is going to be more environmentally friendly. Why? Because if it's at a lower temperature, we heat it up less. We need to heat it up less, which means we use less energy. And if we use less energy, that helps, right? So this is point 1. And then by using less energy, we use less fossil fuels, okay? So we use less fossil fuels, we lead to less greenhouse emissions, all these things that you've done before, right? And those are what we're going for. We are trying to use less energy so that we can use less fossil fuels and release less emissions, okay? And that's basically what that question's asking. Honestly, as long as you talk about list point here, and then one of these other two, whichever one you prefer, then you'd get those 2 marks, okay? If it was a 3-mark question, you'd probably want all 3. All right, neither detergent works well at 60 degrees Celsius. Explain why. Now, we saw this in the RPA as well. When we went to 80 degrees Celsius, the enzyme didn't break down the starch. Why? Because it was, and the fun word to write here is the enzymes were denatured, okay? They were denatured. Always use that word. If the temperature is too high, enzymes will denature. So that would be one of those 2 marks. And what does denatured mean? It means the enzyme breaks down or changes shape. And if it changes shape, it means that there's no active site, or the active site changes shape. No active site anymore. Okay, and so the substrate or the enzyme no longer works. Substrate cannot fit, so the enzyme don't work. Done. All right, enzyme won't work. And that's literally what it is. The enzyme is denatured, so the active site changes shape, so it doesn't work. Question done. That's the key question, guys. That's what you need to be able to do sort of from that Enzymes RPA. I'm about to go into a second question. For those of you that are interested, please hang around. For those of you that just wanted the key part, then you can stop now. All right, so this one's a little bit more challenging, but I wanted to try and give you a bit of an idea about how we handle these sorts of questions. So we've got another enzyme, catalase, which is found in many different tissues in plants. It speeds up the rate of the following reaction. Hydrogen peroxide broken down into water and oxygen. Oxygen being a gas, right? And so we've got this broad bean seedling here. We've got stem, leaf, seed coat, seed, root. So some students investigated whether different parts of the bean seedlings contain different amounts of catalase. Okay, so we put hydrogen peroxide in 5 test tubes, added different parts of the bean seedling to each, and recorded the results after 30 seconds or half the minute. If there was catalase in part of the seedling, oxygen gas was given off. Okay, so more catalase, more cata, all right, more oxygen given off, right? When oxygen is given off, foam is produced. So more cata, more oxygen, more foam. All right, so let's have a look at the results that we've got here. So tubes before adding the plant are these along here. Let's call them 1, 2, 3, 4, 5. Okay, then they've added the various parts here and then looked at the foam. Okay, the student made the following conclusions. Most of the parts of a bean seedling contain catalase. The seed contains a lot of catalase. Stems and roots have quite a lot of catalase. The leaves have a little bit of catalase. The seed coat has hardly any catalase. The student teacher said that the student's teacher, said that the students needed to improve their investigation in order to make valid conclusions. All right, so what's wrong with this experiment? I'll give you a minute to have a look at that picture and just think about it, right? What's wrong? Why can't we compare these test tubes fairly? What's going on? So hopefully, I was thinking put some elevator music in the background there. Most of you will have hopefully seen that, look at how much hydrogen peroxide is in each of these test tubes, right? So there's a different amount of hydrogen peroxide in each. Different volumes, as we would say, different volume of hydrogen peroxide in each test tube. So it's not comparable. Maybe the root only produced this little amount of froth or foam, not because there's any less catalase, but because there was just less hydrogen peroxide to break down when compared to something like the seed, which had, over twice the amount of catalase. Maybe it would have foamed up just as much. So how would we make it fair? We'd make sure that each of these solutions, each of these had the same, I'll try and draw it perfectly here, the same amount of hydrogen peroxide in each test tube, okay? Which would then at least mean that we could compare the results more fairly. So number one, what would I do? I'd make sure they had the same volume of hydrogen peroxide in each test tube. Okay, good. So number one, this is trying to spot experimental mistakes here, which is a skill in and of itself, so it can take a bit of work. All right, so the next one is this. I'm hoping you guys can spot, look at the different sizes of these different cuttings, right? And so this is a really big seed, but it's a really small chunk of root. And it's a fairly small bit of stem as well. And so it looks like they've put in different size pieces of different parts of the plant, right? And maybe because the seed's so huge, it just has more catalase in it than a tiny little piece of root. Maybe the root has more catalase per unit volume, but it's just a different size. So making sure all the pieces that were put in, if you have a seed which is this size, then you need to make sure you put down a root. You put root in, which is the same size as well, okay, in order to be able to compare those results to make valid conclusions, all right? Those are probably the big two flaws that I would see with this experiment, which leads me into my final part. I want you to practice observing those sorts of things because it's common in those methodology questions. All right, so if you were ever asked to write a method like they've asked you to do here, please, these are my 5 steps to success, if you will. The first thing you need to do, and please tattoo this on the back of your hand or forehead, whatever you want, guys. Think, if they ask you to write a method for an experiment, can the person who is reading this do exactly what I do? If they can't do exactly the same experiment you're doing or that you're writing, then the method isn't specific enough. You need to include volumes. You need to include amounts, things like that, okay? 2, what is your independent variable? What is the variable that you are changing? 3, what is your dependent variable? What is the variable that you're going to measure in the experiment? Include all of these in your response. 4, what controls would you use? i.e., what would you keep the same so that you can compare the results? So controls here would be exactly what we just talked about. The same volume of hydrogen peroxide in each of the test tube, control number 1. The same mass of parts of the plant, whether it's seed, stem, etc. Control number 2, because then we're only changing the concentration of catalase in each of those parts. Okay, so make sure you have controls which allow you to compare your results. And lastly, provide units. Don't say add a hydrogen peroxide, say add 5 mils of hydrogen peroxide, add 5 grams of seed, right? Just make sure you say and provide your units for the experiment. If you do these 5 things when writing a method, you're going to get so many more marks than you ever would if you ignored them. Okay, guys, I know this is a bit long, but I want this to try and I want to run you through my process of thinking when I do these, and I'm a little bit slow, so naturally it takes me a little bit of time to run you through my thinking. I hope this helps, guys, and I'll catch you later.